Question number 17. A giant chromosome with a number of chromonimata is A. Lambrusch chromosome, B. Heterochromosome, C. Polytene chromosome, D. Supernumerary chromosome. Whenever we are dealing with relation to chromosomes, if the chromosomes are large and they go to form a sort of a reticulum, if the chromosomes are large and they form a sort of a mesh-like structure, this mesh-like structure is termed as chromonimata, and they are large, hence they are termed as giant chromosomes. So, giant chromosomes with a sort of a chromonimata, because there are many threads formed over here, many threads which are wound, it is termed as polytene. So, polytene chromosomes are nothing but large giant chromosomes. So, a giant chromosome with lot of chromonimata is otherwise termed as, answer is A. Lambrusch chromosomes, B is heterochromosomes, C is polytene chromosomes, D supernumerary chromosomes. The right answer is C, which relates to polytene chromosomes. Answer is C. Question number 18. In DNA replication, the formation of a new strand on the DNA template can be initiated only if dash is present. A. DNA polymerase 3, B. DNA primer, C. RNA primer, D. DNA polymerase 1. If we have to, there is a need for the formation of a new strand on the DNA template, the DNA template is called the master molecule. On the master, if ever we have the presence of an RNA primer, a primer is a start molecule. It is necessary that an RNA gets attached to the DNA. Then only we have the formation of the proteins, and again it can give rise to the new strand. So, whenever a new strand has got to get formed, an RNA primer adds. So, this one, a primer is a starting molecule or it adds to DNA replication. Hence, because it adds and there is a synthesis of an RNA strand and an RNA DNA complex gets formed, that is why it is called an RNA primer. So, among the choices, the right answer is RNA primer. That relates to answer is C. Question number 19. The sites or points where crossing over of homologous chromosomes of a pair occurs are termed as A. Chromomeres, B. Centromeres, C. Chromocenters, D. Chiasmata. During crossing over, if during meiosis, at meiosis, wherever there is crossing over, we are dealing with reciprocal exchange of non-homologous chromatids of homologous chromosomes. So, whenever there is crossing over is indicated by the figure X. When you look under a microscope, you find that chromosomes can be varied. It can be like an L, or it can be like a J, or it can be like a V. The points where a genetic exchange of chromatin material occurs, these X-shaped configurations or the points of exchange where there is exactly reciprocal exchange of the genetic material. This is the point area where there is reciprocal exchange of the genetic material. This is termed as chiasmata. The right answer is D. Question number 20. Triticum durum is A. Diploid, B. Triploid, C. Hexaploid, D. Tetraploid. When you take wheat varieties, In wheat, there is a specialized variety which is termed as Triticum durum. Now, Triticum durum is hard wheat. It is used in Italian foods like pasta, and its gluten content is less. So, it makes hard wheats which is also made with relation to certain compounds called macaroni food. So, macaroni food and pasta is primarily derived from Triticum durum, and Triticum durum is one of the sort of wheat varieties which has been improved by means of mutation breeding. And it is an example of polyploidy, where exactly the chromosome number is tetraploid, that is 4 times. The right answer is D. So, hard weeds are primarily termed as Triticum durum, and it is a tetraploid. Question number 21. The nucleosome core has A, H1, H2A, H2B, H4, B, H2A, H2B, H3, H4, C, H2A, H2B, D, H1, H2A, H3 and H6. When you take a nucleosome is an important core particle of the chromatin. The chromatin material of the DNA is organized into a nucleosome, and the nucleosome predominantly is made up of H2A, H2B, it is made up of H3 and H4. Again, it is made up of 2 molecules each of the above. So, you have got 4 molecules into 2 is equal to, it goes to form an octant, 8 molecules. So, the 8 molecules of the nucleosome primarily acts like a sort of a spool. So, around the nucleosome, which is made up of 8 particles, 4 on one side and 4 on the other side, the DNA is bound, and 2 nucleosomes are connected. Nucleosomes are connected by a linker DNA. So, a nucleosome is primarily a sort of a packing material or the spool, and the core is primarily made up of 8 molecules of, that is 2 molecules each of H2A, H2B, H3 and H4. The right answer is B. So, a nucleosome facilitates compact packing of a sort of a large DNA. A large DNA is spun round nucleosomes, and many nucleosomes together go to form a sort of a solenoid. And the solenoid is just like a sort of an electric cable, wherein all the particles of nucleosomes are wound just like matchboxes. If you have 500 matchboxes, 500 matchboxes can be wound together to form a sort of a sheath which is termed as a solenoid. So, DNA protein packing primarily occurs by nucleosomes, and nucleosomes is in turn made up of histones. Histones are the basic packing materials. Question 22. Linkage map is associated with A. Neurospora, B. Drosophila, C. Pisum sativum, D. Chlamydomonas. Now, the meaning of the term linkage. Linkage is a tendency of 2 genes to be inherited as an unit during genetics. So, during linkage, 2 genes are held very close to each other and they are inherited as an unit. Each unit of inheritance which shows linkage can be categorized and placed on a map which is termed as a linkage map. Now, in a linkage map, which is a diagrammatic representation of places on the sort of a DNA where linked genes are present, that is, it may involve an A gene and a B gene, a cluster of A and B, or it may include another cluster called M and N. So, this process of arrangement of the linked genes diagrammatically is termed as a linkage map. Linkage map has been studied by Morgan, and it has been studied in Drosophila melanogaster. Drosophila melanogaster is otherwise termed as a fruit fly. Drosophila melanogaster is also called the Cinderella of modern genetics, because all studies on linkage as well as crossing over has been studied with Drosophila. So, in the case of the Drosophila, due to linkage, we have the development of different shapes of the Drosophila eye and also the color, wherein this has been the first studies with relation to linkage. So, among Neurospora, Drosophila, Pisum and Chlamydomonas, the right answer is B, that is Drosophila melanogaster. Answer is B. Question number 23. The DNA unwinding protein as well as the place where the single strand DNAs bind are termed as A. Replicon and Okazaki fragments, B. Polymerase and topoisomerase, C. Replicase and replication fork, D. Helicase and DNA pol 3. The place during DNA replication. During DNA replication, there are several enzymes. The DNA unbinding protein is termed as replicase. Replicase is the unwinding protein. If we have two strands of the DNA, the two strands are separated, there is unwinding. It is just like you open out a thread. So, the unwinding enzyme is primarily termed as the replicase. Replicase is the unwinding protein, and the place where the DNA strands are bind, the place, this is the region where the single strand regions bind. The location where it binds is termed as the replication fork. So, the replication fork is Y shaped or it is Y shaped, and it is the place where both the single stranded DNA come together. So, the region, this region is primarily termed as a replication fork. Now, in addition to this, there are some sort of enzymes which are termed as primase. They are the primers, and again we have got the DNA polymerase, pol 1, and again you got DNA pol 3, which are primarily involved in zipping the sort of a DNA as well as joining the sort of a ligases, joining the small pieces which are primarily termed as Okazaki fragments. So, the most important enzymes for unwinding as well as formation of the replication fork are replicase and replication fork, and the right answer is C. Question number 24. When adenine is replaced by guanine, it means A. Base substitution, B. Transition, C. Transversion, D. Point mutations. Whenever we take mutations, in mutations it can either primarily mutations can involve shifts. There is a change, the change in the sort of a nucleotide can involve shifts which can either involve purines with purines or pyrimidines with pyrimidines or purine with pyrimidines. Whenever a purine gets replaced by another purine or a pyrimidine gets replaced by another pyrimidine, like in the example, if adenine is replaced by guanine, adenine is replaced by guanine, then this sort of a change is primarily termed as transitions. So, transition type of change in the nitrogen bases is very common. So, transitions involve purine to purine or pyrimidine to pyrimidine. Hence, this process is termed as transitions. Whereas, when we take transversions, in transversions, a purine can be substituted by a pyrimidine. And in a base substitution, one nitrogen base is replaced by another nitrogen base. A point mutation refers to a single nucleotide change. So, among the choices, the right answer is transitions, that is, answer is B. Question number 25. The specific point where polytene chromosomes appear to be attached together is termed as A. Centromere, B. Chromomere, C. Centriole, D. Chromocenter. When we take polytene chromosomes, primarily from the word polytene, you understand polytene means many. So, the chromosomes are just like colored ribbons, many colored ribbons. So, the specific point where they are all attached together, this particular point is termed as chromocenter. So, the chromocenter is a central point in a polytene chromosome where the chromosomes appear to be attached together. The term centromere relates to the attachment in the case of a single chromosome, the point of attachment. Chromomere primarily relates to heterochromatin and euchromatin. Chromomere relates to the point of heterochromatin and euchromatin, that is the colored regions of the chromosome. Whereas the centriole relates to animal cells, and the right word here is chromocenter. Chromocenter relates to polytene or many chromosomes. Hence, the answer is D. Question number 26. Polytene chromosomes result due to A. Mutations, B. Non-homologous, C. Endoduplication of chromosomes, D. Somatic pairing of homologous chromosomes. Now, why is it that chromosomes can increase in their number? Chromosomes increase in their number because there is a need for higher chromatin content, and the higher chromatin content is a specific characteristic feature of the single sort of a species. So, within a single species, if the single cell can endoduplicate, can again form various chromosomes of the same order, it is by the process of endomitosis. By endomitosis, there is endoduplication, that is when there is doubling of the chromosome content. It primarily is a result which leads to the formation of polytene or many chromosomes. Hence, among the choices, C, endoduplication of chromosomes, is the right answer. The answer is C. Question number 27. A puff and a band are seen as A. Balbiani rings, B. centromere, C. polysomes, D. autosomes. Whenever you take Balbiani rings of giant chromosomes, in a giant chromosome, the giant chromosome exists in the form of a band. So, these are primarily termed as bands of a giant chromosome. They are regions of heterochromatin and they are dark staining. During activity, that is when RNA synthesis has got to occur, the same bands will puff and this region is termed as puffing. Puffing indicates RNA synthesis and it indicates activity. So, bands and puffing constitute together the Balbiani ring, which has been studied by the scientist whose name is Balbiani in salivary gland chromosomes. Hence, the answer is A. Question number 28. The mode of DNA replication is semi-replicative. This has been proved by A. Watson and Crick, B. Hargobind Khurana, C. Taylor, D. Michelson and Stahl. Whenever we take DNA replication, during DNA replication, we have two strands. The two strands are the parental strands. The parental strands unwind. Then we would have two single strands of the DNA, one from the paternal line and another from the maternal line. When the two strands separate, they in turn can synthesize two daughter strands. One on each. If there is an A strand, if this is the A strand and this is the B strand of the DNA, the A strand can synthesize the daughter strand which is termed as A dash, and the B strand can synthesize another strand which is termed as B dash. So, by using radiothymine, scientists Michelson and Stahl had conclusive evidence to prove that the DNA replication is semi-replicative. Semi-replicative means half the characters of the parents are always conserved. So, always whenever DNA reproduces, 50% of the characters are attributed to the parents and 50% of the characters are attributed to the offsprings. Thereby, in every generation, the parental character is transmitted. So, the transmission of the characters wherein 50% of the characters are from the parents, this method of replication is termed as semi-replicative method. So, in semi-replicative, 50% of the characters are from the parents and the right answer is D. Question number 29. Embryo and endosperm of rice formed by crossing a tetraploid female with a diploid male can be A. hexaploid and triploid, B. triploid and pentaploid, C. triploid and hexaploid, D. diploid and haploid. Whenever we take rice, in rice we have the embryo as well as the endosperm. So, if there is a crossing between a tetraploid female it combines with a diploid male, then in most situations what happens is we have 4 plus 2 is equal to 6n. 4 plus 2 is equal to 6n. Now, when you take the embryo because it is 6n, it is reduced, it becomes 3n. Embryo becomes 3n, and here in this case, if you take the endosperm always has a primarily it has got a 5n. So, thereby here this region this is termed as pentaploid and this is termed as triploid. Thereby among the choices we have B, that is triploid and pentaploid as a right answer, and the answer is B. Question number 30. When two gene loci exist as cis and trans but produce identical phenotypes, they can be designated as A. part of the same gene, B. different genes, C. pseudo alleles, D. multiple alleles. When we have two gene loci, on one side we have capital A and capital B, and we have a small a and a small b. This is called cis. And on the other side we have a capital A and a small b. We have a small b and a capital A, capital B. This is termed as trans. With relation to genes, we are dealing with relation to gene loci. Gene loci can exist as A, cis, or B as trans. So, when two genes exist in cis and trans, but either way they produce the same or they produce similar phenotype, irrespective of whether it is in cis or in trans, they produce the same sort of a similar phenotypes. So, these alleles, a pair of contrasting characters is termed as alleles. These alleles are termed as pseudo alleles. Hence, the answer is C. Question number 31. Angstrom is equal to A. 0.01 millimicrons, B. 0.001 millimicrons, C. 0.001 millimicrons, D. 0.401 millimicron. Whenever we take angstrom unit, an angstrom unit is given as equivalent to 0.0001 millimicrons. This is the right choice. Hence, the answer is C. Question number 32. Point mutation means A. Alteration of the DNA sequence, B. Deletion of a segment of the DNA, C. Change in the single base pair of the DNA, D. A gain of a segment of a DNA. Whenever we are dealing with point mutations, point mutations are specific. They are changes with relation to a single base pair of the DNA. So, if a single base pair, there is an alteration in a single base pair of the DNA, it is termed as point mutations. So, the right answer is C. Question number 33. In a cell cycle, DNA replication occurs during A. G1, B. G2, C. S, D. Mitotic metaphase. Whenever we are referring to DNA replication, replication relates to increase in the DNA content. Parallelly, also increase in enzyme activity and also the synthesis of new proteins. So, all increase, quantitative and qualitative increase in the DNA primarily is attributed to the S or the synthetic phase. Or the synthesis phase of cell cycle. So, DNA replication occurs during the S phase, that is, the answer is C. Question number 34. The basic proteins of the DNA are A. Albumins, B. Histones, C. Non-histones, D. All the above. When you take DNA, the deoxyribose nucleic acid is a genetic material, and the genetic material is a long molecule. It is a linear DNA molecule. The linear DNA molecule is always held or it is compacted by proteins which are basic in nature, and these basic proteins are primarily termed as histone proteins. So, the histone proteins they act like clips and they bind the sort of a DNA. If when a DNA is a long molecule, the DNA is wound and it is held by means of the histone proteins. So, histone proteins just act like clips by means of which you can hang the clothes. So, similarly histone proteins, they are binding proteins. They are basically binding proteins and they are involved in compacting the DNA as well as they are involved in the packing of the DNA into the sort of a chromosomes. So, the answer is B, relating to histones. Question number 35. How many mitotic divisions are essential for establishing 128 cells? A. 5, B. 7, C. 14, D. 10. Imagine we have mitotic division. A mitotic division is also termed as equational division. In an equational division, one cell divides to form two cells. If we had 128 cells, 128 would have got from 64 cells. 64 cells you could get it from 32 cells. 32 you could be getting from 16 cells. And 16 cells you could be getting from 8 cells. and 8 cells again from 4 cells, and 4 from 2. So primarily when you look at the sequence, we have 1, 2, 3, 4 and 5. We require primarily 5 important type of mitotic divisions, so that it increases like from here, it increases so that we get 128 cells. So in order that we get 128 cells, there is the need for 5 mitotic divisions, hence the answer is A.